Hello everyone, welcome back to another video lecture of the series that sexual reproduction in flowering plants. Now right after this is almost we are reaching towards the ending portion of this chapter, that is the fertilization events have occurred. Before that, pre-fertilization events, pollination, everything have occurred. Now post-fertilization changes are also done. Now let's see that what is the basic difference between the embryo of monocotyledonous plants and dicotyledonous plants. As we know that angiospermic plants are of two types according to how many cotyledons they have. Dicot means that two cotyledons are present and monocot means only one cotyledon is present. So of course the angiospermic plants are also very much advantageous because they can produce the seeds, and as the seeds are much more viable in nature than any sort of other modes of reproduction, that is why it is said to be much more advantageous over any other kind of plants, right? So now seeds can be of also two types. If the seeds are naked, then we can consider it as the gymnospermic characters, and if the seeds are closed within the fruit, which is the characteristic feature of angiosperms, and here we are talking about angiospermic plants. So let's see that what are the embryo and how the cotyledonous development, everything is differentiating from the monocot to the dicots. In case of dicotyledonous embryo, we can find that there are the two cotyledons. These two are the cotyledons, okay? And in the cotyledons, as you can see that among the cotyledon there is one axis, and if we are talking about the upside portion of the cotyledon, that is upper portion of the cotyledon, that portion can be considered as epicotyle, and the lower portion of the cotyledonous axis, that is considered as hypocotyle. So upper portion is considered as epicotyle, from which region it can give rise to the plumule. Plumule is extremely important to give rise to the shoot, that is stem, leaves, everything will grow from the plumule region only, and the hypocotyle will give rise to the radicle. Radicle is also very important because from radicle the roots will be developed. Now in few species you can find the root cap, but in few other species we cannot find the root cap. Instead of finding root cap, this is found in most of the aquatic plants. Instead of finding root cap, they are having air pockets, okay, so that they can float very easily in the water. So air pockets are present in such cases, okay. So that is the region from where the root cap can be formed, and this is the radicle. Radicle can be formed from the lower portion of the cotyledons, okay. That portion is considered as hypocotyle. So from the hypocotyle region the radicle can be formed, and root cap is present in the outside portion of the radicle. And as you can see that the number of cotyledons are 2, that's why this is considered as dicotyle or dicot embryo. Now let's move ahead into the monocot embryo. What we can see is, in case of monocot embryo, in the cotyledon, there is only one cotyledon. The cotyledon is spelled, special name is given to that, that is scutellum. That is, what is the name of the monocot cotyledon or monocotyledonous cotyledon? That is scutellum, okay. What name is given to that? Scutellum. So scutellum is the name of the cotyledon of the monocot embryo. Next come to the hypocotyle and epicotyle. As we know that epi means upper, so once again the upper cotyledonous axis is considered as the epicotyle region, and the lower cotyledonous axis, that is considered as the hypocotyle region. So from the epicotyle region, once again the shoot apex will be formed, and from the hypocotyle region once again the radicle and root caps will be formed. Now two extra portions are present here which is absent in dicot embryo. Here you can see that shoot apex region is covered with the extra cap-like structure. Can you see here a cap-like structure present here? So this cap-like structure is called as coleoptile, which is covering the shoot and or the shoot apex, and the portion cap-like portion which is covering the root or the radicle, that is called as coleorhiza. So what are the three extra features? Scutellum is extra, coleoptile is extra, coleorhiza is extra. Coleorhiza means root, so the root covering is considered as the coleorhiza, and coleoptile means the shoot covering. Okay, and one more important thing, that is epiblast. What is this? This is nothing but another dormant kind of cotyledon. That means this is not fully grown. This is also another structure, but as you can find that this is monocot. That means only one cotyledon is present. The second cotyledons which was to grow but which has stopped in the midway. Its growth has been stopped. So it has become dormant and it is not at all growing, and that type of structure is considered as epiblast. It is very important to remember the epiblast in this case. Okay, so in case of monocotyledonous embryo, we can also consider this type of part as rudimentary part. Okay, we can also say this part as rudimentary. Let me write it down here. We can also call this part as the rudimentary part or rudimentary cotyledon, which is termed as epiblast. So this is all about the dicot and monocot embryo. In my next video, I am going to talk about apomixis and polyembryony, which are the last two topics of this chapter. And here I am wrapping up my video. I hope that this video is helpful, and if that, then please don't forget to like, share and subscribe to my channel. Thank you.